3,
Hello,
2.
everybody! Welcome back to Fossil Friday Chats. We are back on the air after a lovely summer break. I'm Brittany Stoneberg from the Western Science Center. With me, as always, is my co-host, Gabriel Santos. And joining us today is somebody we are super excited to have on the program and to continue our general love affair with the world of plants and paleobotany. Aviwe Matiwane, so nice to see you.
Hello, thank you so much for having me on your program.
Oh, we are so excited to have you. As we were talking about before we went on the air, our new goal with Fossil Friday Chats is to get everybody to fall in love with paleobotany.
Yes, because it's the best thing ever.
Exactly. That is our mission.
Absolutely.
That's our new mission statement. We're rewriting everything. It's just paleobotany from here on out.
Definitely.
We've been trying to get Aviwe on the program for months, and so we are super excited to finally have you be able to join us all the way from South Africa. Oh, one moment.
I think we might have frozen.
Oh,
Just
no!
a second.
Yeah.
There we go.
We have like a huge time difference, and I'm so envious. Can you hear?
There you go.
Yeah, you cut out just for a second. You're fine.
But we got you back.
Oh, okay, sure. Like I said, yeah, the weather here is a bit bad, so it's actually affecting my network and connection, so I apologize.
Oh, that's all right.
No.
We will carry on. All right. If you're not familiar with Aviwe Matiwane's work, she is a paleobotanist and ecologist from South Africa. She's a lecturer at Rhodes University, a research associate at the Albany Museum, and an avid science communicator, which is how we came across her amazing work. She primarily works on Permian flora from Gondwana, plant biodiversity in southern African forests, has an interest in Triassic flora, and recently ventured into the field of STEM and Academic Literacies in higher education. That is all really cool.
Yeah, amazing stuff you've been doing.
All right, everybody. So as always, if you have questions for our speaker, go ahead and put those in the chat. We will do our best to get to as many as we can after her presentation. And Aviwe, whenever you are ready, go ahead and share your screen and tell us all about why plants are the absolute best.
Sure. Can you see my screen? Okay. So thank you so much for having me again. I'm here to share my work today, and I hope to make sure that everyone falls in love with plants after my presentation. So like Brittany had I said, I'm a paleobotanist from South Africa, and I am now a full-time lecturer at the Churchill department, which is a Center for Higher Education in Rhodes University. I have a passion for animals. I love animals. As you can see, these are all my dogs and rescued. And this is what I love doing. This is one of the things that actually brings me joy. And I am from a rural village called Mqanduli, which is in the Eastern Cape in South Africa, the Wild Coast. I'm sure if you go on Google and you Google the Wild Coast, you'll see how beautiful my country is and my part of where I live is. And yeah, so this is me, this is my existence apart from being an academic at Rhodes University or science communicator. This is the university where I am based. This is Rhodes University. My department is actually on the other side, but these are very iconic bicycles that are, well, it's a structure that's in my university. And this is my department, the botany department at Rhodes University, where I'm actually doing my PhD. This is the Albany Museum, and I'm part of the Earth Science department at the Albany Museum under the supervision of Dr. Rhodes Previck. And so I've always loved plants and nature from a very young age. So for my master's and my honors, I did work in forests. I did a forest biodiversity, and I was looking at the distribution of plants along the escarpment of the Great Escarpment in South Africa. So I had 26 sites that I was sampling, and I looked at the diversity across a moisture gradient. So this site being the most dry area in South Africa, and then along the gradient with Manzimnyama at the end, which is much more moist. I will not go into detail about this research, but there's a paper coming out that I'm sure if you were interested in, you'd be able to read up on it, and I hope you will enjoy it. So just pictures of some of the patches that I actually worked in. So this one is in Tsolo, somewhere in the rural Eastern Cape. There's a cow over here, Nguni cow, and some of the huts from the local village. This is one of my favorite forests. This is Maiden Dam, and I used to sample there, and this is in Boschberg in Somerset East. And over here we have Gulandoda, which is in the Eastern Cape. This forest had a huge snake, and so the villagers were actually quite scared that we were sampling in this area, but we didn't know. We only found out after we were done. So, so yeah, I can tell you stories, but this was one of the the starting stones of actually turning me into a paleobotanist, because I fell in love with taxonomy from there, and from identifying all these studies that I was to gain knowledge in taxonomy and gain knowledge in plants. So after my master's, I was stuck, and I was, before actually submitting my master's, I was sort of in a loop. I didn't want, when you have writer's block and you're so tired of writing the work, your research, you just want something new. You just want to start something new. I started volunteering at the Albany Museum, and through my volunteer work, I met my now supervisor, Dr. Rhodes Previck, and she welcomed me into the field of paleobotany and also exposed me to the field. She took me to trips. Field trips with her, and it just went on like that, and it was absolutely amazing. So these are some of the plants that we found there. As you can see, the orchids everywhere. They were very beautiful. Absolutely. Yes, some of the pictures actually switched. So some of things that we found in the forest that I was working on. So why study paleobotany? So I had a vested interest in nature, like I said before, and the whole fact that you're discovering new things. You're the very first person to find the fossil, you're excavating in the spot, after millions of years of it being underground. And the whole thing of finding something new and contributing to science just gave me, in terms of diversity, I also wanted to inspire people from the same background as me to actually join the field and be interested in the field. So this is a picture of the 3 paleobotanists in Africa. This is Professor Marian Bramford at the at Wits University, Evolutionary Studies Institute. This is my supervisor, Dr. Rhodes at the Albany Museum, and then this is me. So as you can see, there is a huge need for paleobotanists in our field, particularly studying a macroflora in Africa. So I hope this also, if people are watching, can see that we are in need of more paleobotanists in order for us to actually progress our science and to catch up with the rest of the world. So this is the team that I work with. Sorry, I just need to drink. I'm sure some of you know Professor Bruce Ouberg, and some of our team members from various parts of the world. There's Dr. Michael Day from England, and Barry from Brazil, from Argentina. And so this is the lovely team and some of the other people that we also have in our team. And so we also have volunteers. So we have Tanya Paul Mullins, who is a world-renowned artist, and she does a lot of collaborative work with us. There's a lovely exhibition that she did that was a traveling exhibition that she did of women in science, and I'll share some of the links with you later of the things that she was doing. So South Africa is very well known for its therapsid fossils, and there's a lot of work that's been done on them, and and a lot of people want to work on these fossils, and they're very amazing, and they do show a progression through time of the evolution of these therapsids in that actually occupied and stayed in the Karoo Basin of South Africa. And South Africa is also very well known for its dinosaur fossils. We have the oldest dinosaur embryos in the world, the oldest evidence of repeated use of a nesting site, oldest evidence of a colony of nesting in dinosaurs, and these are found in the Golden Gate National Park in the Free State in South Africa. So let's get to the more interesting things, the plants. So I work with a plant group known as Glossopteris. The Glycopteris name was first, installed in 1828. This is an example of the leaf. It is tank-shaped, has a prominent midrib, and has net venation. It is abundant as an impression fossil in South Africa, and it is synonymous with the Permian of Gondwana. So the reason my my talk was known as, I decided to use black gold, is because the Glossopteris actually form the oldest coal deposits in our country. And they contribute a lot to our economy, because coal, unfortunately, it's a double-edged sword, because it also contributes to climate change. But for us, I love working with it because this is what this is what the plants that the fossil plants that I'm working on, created. And in South Africa, the oldest coal deposits are found in the northern parts of the country. And this is just a zoomed in picture so you can see. So this is the Karoo Basin, the world famous Karoo Basin. And as you can see, so it goes according to age. So you have the oldest, and then as you go towards Lesotho, these are the youngest deposits. So this is a reconstruction that was done by my supervisor, Dr. Rhodes University, and it's a reconstruction of coal swamps in South Africa from the Middle Permian. And this is an example of what Glossopteris might have looked like. So Glycopteris is thought to have been a plant that is about 30 to 35 meters tall. And this is evidence found from Antarctica from polymerized material. And there is, I mean, my supervisor does the best reconstructions, and there's evidence of plant damage, which you'll see at a later stage when I actually show you some of the wonderful pictures and material that we've collected from my sites. So these are coal-associated Glossopteris leaves. They're huge, massive, and absolutely beautiful. I think South Africa is only rivaled by Australia when it comes to great fossil material, but I think we're still the best. So the continental drift, and as you can see, it was found, since it was found in Africa, South America. Sorry, there's some noise in the background. I'm not sure if. Yeah, okay. So it was found in Africa, South America, Antarctica, India, and Australia. And so because it was found in all these in all these land masses, it was used as evidence to support the theory of continental drift. So Glossopterids are quite a diverse group. So you have the tree. This is just a brief, a generalized reconstruction of the plant. And with botany, paleobotany, each plant actually each part of the plant gets a separate name or genre, even though it comes from one plant. And this is due to the fact that certain, elements are discovered first before others. So, for example, you have a pollen structure, but when the pollen is actually within the combined structure, it's known as arboriella. When the arboriella clusters are actually attached to a scale leaf, they are known as erythmonia. So, and when erythmonia is attached to a cone, it's known as something else, such as squamella, which I'll talk about later on. So this is where my research comes in. As you can see from the previous slide that I showed you, the species identification has proven to be very challenging for this group. So there has been a number of approaches that are used in classifying Glossopteris leaves and Glossopteris, elements. So Glossopteris taxonomy is highly subjective. So the fossil record is highly fragmented because there are few conclusive links between the reproductive organs and the leaf type. So the leaves are morphologically conservative, yet they show a high degree of intraspecific variation. The leaves are also found in far abundance than the fertile structures known as the fructifications. So the fertile organs are the most taxonomically informative, however, they're very rare. So their use in it is quite important for us to understand the taxonomy of this group. And like I said, the affinities of this group are actually quite controversial and have been for a number of years. This is a 200-year-old problem. So morphological studies of the fertile structures indicate that Glossopteris exhibit a high degree of provincialism. There are 20 species of fructifications within South Africa, and very few can be positively identified beyond the southern African superregion. So what does this mean? This implies that broadly applied Glossopteris leaf names are probably misleading. And this has probably led to false biogeographic and false photogeographic conclusions. So the Glossopteris leaves have a potential to be useful indicators for temporal change in the fossil record. So their use of these leaves as a fossil, as an index fossil in geological units within the Permian has been lacking due to a consensus as to the best way to classify these leaves, so how to describe them, due to a general lack of faith that the species can consistently be discriminated. So where does my research come in? So new approach acquired or required. So the Glossopteris leaves need a standardized approach, taxonomic approach globally. So my study basically addressed these contentions by taking a morphometric approach towards species identification. So my study is actually an exploratory study that uses stats to identify which features offer the highest degree of useful and consistent discrimination between species. So how was this achieved? So this was achieved by observing whether morphological variation in Glossopteris leaf populations can be used as a proxy for species identification, and how do the cluster boundaries change based on species, based on characters used to define them, and whether it is reasonable to assume that morphological clusters represent a species. So my current study assessed a pattern of morphological variation within two populations of Glossopteris leaves and determined new morphological features that could practically be used to discriminate between morphospecies, and also described a new flora from Ouberg Pass in Sutherland in the Northern Cape province of South Africa. So as you can see, this is Glossopteris. It is actually a simple leaf with very few features. And in my study, I had to work with these, photograph hundreds of these leaves, do measurements on them, and just try and find a good way to add 100 plus specimens. So I only selected leaves that were 60 to 100% complete. I identified 58 features. So these features consisted of features that I found in the literature, and also features that I thought were also important, that needed to be included in my work. So these features included, for example, the apex angle, the mesh width, mesh length, the angle of the veins, and so in different parts of the leaf. So the leaf was actually divided into three main parts, and then subdivided into three other parts: the proximal, which is close to the midrib, and the midlaminar, and the marginal. And then from there on, there were certain features that were measured within them. So there were not only quantitative features that I measured, there were also qualitative features that I looked at. So these were the descriptive features, the shape, for example, of the mesh, the shape of the leaf, the shape of the base, and the features are related to the midrib, etc. So in South Africa, as you can see, most of the work has been done in the northern and eastern parts of the country. So we know a lot about these fossil records and occurrences this side, but very few has been found in the southern part of the basin. So if you look in the southern and the western part of the basin, so if you look at the records of the Middle Permian, so you only have two localities over here that are suggested to be of a Middle Permian origin. but they're only based on fragmented leaves. So it could be like it's an occurrence. So where you have one or two leaves that are not really identifiable to to an extent, and also it was not, these areas are actually not as conclusive as the areas I'm about to talk about. So we identified two areas for my study. So this is Sutherland over here, and it's in the Northern Cape province of South Africa. And there was also Gwayaya, which is in the KwaZulu-Natal province in South Africa. So both sites are in different parts of the basin, and also the sites are different in age. So let's start with Gwayaya. Gwayaya is, I'm sorry about the phone. Gwayaya is a locality in KwaZulu-Natal, which is a late Permian of age, and it is actually one of the sites that I love because you'll, as we go on, you'll see why. It is part of the Beaufort Group, which is equivalent to the Normandy Formation, and it is from the Upper Permian. So this is why I love this site. It has the best leaves, and the preservation is absolutely amazing and stunning. Previous work that was done here identified over 10 morphospecies, and with my work and my reevaluation of the actually us reduced the number of morphotypes to 4. And then within those 4 morphospecies, we have subtypes as well. So these subtypes are basically the leaves that actually show similarities within each other, and but there are differences within the different subtypes, but not all the morphospecies have subtypes. And then there are Glycopteris fertile structures that are found. For example, we have and we also have red beer. So and we also have I don't do any field work site. The work is actually based on fossil collections years ago. And also, yeah, this is the only site that I wish I had actually visited. But I mean, as you can see, the material is absolutely stunning. And yeah. So the other site that I worked on is Ouberg Pass. Now, this place is amazing. This is a beautiful, beautiful area. And whenever we go there, I mean, as you can see from this picture as well, it is absolutely majestic. I mean, you can take all landscape and so it's actually located just outside Sutherland, and I will show you in the next section. Yes, so as you go down the small plateau, you actually, it's our site is actually located somewhere down here. It is a road, it is along a road cutting, and this is actually a very famous route in South Africa. It's used by bikers, and as you can see, the views absolutely amazing and beautiful. And so our site is somewhere along here, down the road. It takes about an hour to hike down, but I mean, we always prefer hiking down than actually driving down. I mean, well, our team members actually drive down, and then some of us hike down because, I mean, it's just beautiful. It's a beautiful locality. So these are some of our team members, and this is us on the site. We're just excavating layer by layer, trying to get all the lovely fossils and material from the rock. So Ouberg Pass lies in the southern Karoo Basin, in a region rich in fossils of terrestrial vertebrates of the Eodisonodon assemblage zone. A detailed biostratigraphic framework, together with multiple ash dates in the immediate area, provide an excellent context for my study. So this is an example of the ash layer, and there were a lot of zircons that were dated from this area that put it in the Middle Permian. So this is shrimp dating that was done by Lancy et al. 2013 from sites that were actually close to my study site, and there was actually one that was closer by, but I forgot to actually put the actual star to show. And it actually forms part of the Guadalupian, which is a part of the Middle Permian in South Africa. So this is an example of some of the leaves that we find in the site. Absolutely stunning. And the preservation is amazing. And so some of the leaves are absolutely huge. And I mean, we spend the whole day there, and you just scream most of the time whenever you discover something new and different. Unfortunately, the site is not as diverse as Guayaya in terms of leaf morphotypes, but morphospecies. But this is actually a wonderful site because it's the first conclusive Middle Permian site in South Africa. So this is just a plate to show you some of the leaves that we also found. Absolutely beautiful leaves, stunning preservation. And I mean, I had a joy working with these plants. So these are some of the fertile organs that we found. Escortia, Urticaria, Erythmonia, the Erythmonia that I showed you previously, and Squamella. So this is the first record of Squamella outside Australia and Antarctica. And also Legitonia. This is also the oldest Legitonia in the world. So just so you know, most of the flora in South Africa is found in these parts of the country. And then we also discovered another site in Ouberg Pass. And now to add, we have Ouberg Pass and another site, which is Guayaya which is called Onder Karoo, just lower down. Like I said, this is the oldest Legitonia in the world. Legitonia is mostly associated with late Permian floras, and now we have it in another part of the basin and also in a different time period. And these are some of the other floras that were found. We have Chazaneurin, some seeds, and a lot of axes that we can't identify at the moment. And then we have wonderful insects that we found at Ouberg Pass. We have bivalves. There are tons of insect wings and legs, and also insect bodies that you can find in the area. I mean, an insect is always a highlight because you see a lot of leaves, but the moment that you find an insect, you are absolutely excited because it is something new and exciting, but the leaves are the best. The leaves are the best. So this is the first time you actually noted a transition from scale to leaf. And the scale leaves actually have this umbo at the tip of the leaves. Sorry, let me just check. Yes. And you can see the transition, and from not having a midrib to having, for it to actually start developing, and then disappearing at the end of a more mature leaf. And just so you know, so let me just go back. So you see this transition over here. This was actually first noted by Mary White in the 70s. And she was the very first person to describe squamella. Remember, I said squamella is only found in Australia and Antarctica, and we also found it, but we found it in mats. So this is actually quite significant. And we also found cones of squamella, and there actually are cones of squamella that were found, but not in the same structure as we have found them. So Mary White actually had a relatively good idea about what was going on, because she was also the first person to note that this transition from scale to leaf, and this transition is seen in the fossil record, but the reconstruction of squamella is different from what she had drawn or described. I mean, she did absolutely an amazing job with this, to think about the material that she was working with, which was quite minimal. So I'll just give you a summary of some of my findings, and then you can ask me questions later if there's something that you want me to clarify. So we found that the middle portion of the leaf is more informative. And I mean, you'll probably never be able to define the natural species of Glycopteris until you actually find maybe, a branch that has everything, or, different clues that are more complete, in a sense. And also, a broad approach is not applicable. There's just too much species variation. And significantly, the leaves cluster according to locality, and some do cluster according to morphotypes. So there were also different attached fertile structures, and also different leaves that were attached to, associated with different leaves, I mean. So also in terms of description, describing Glossopteris, you can't just describe it with one single feature. There are multiple combination of features that I identified, and the team identified, that actually would be more taxonomic informative, like features including the vein angles and the mesh width. Our paper's actually coming out very soon, so you'll be able to read all about the new discoveries that we found from this site. So the outcomes are basically to understand at one site, but also using comparison with another, which was Guayaya, and also contribute towards a biostratigraphic framework based on adding more localities would actually help us see what if this could be a useful feature in biostratigraphy in the future. And also I've developed a database for this research that is generated, with detailed features that I identified for the description of these leaves. And so we have a site-specific database, and also, it would be also great to have to contribute this database to a global platform, which can be used to allow for comparisons for species, from across Gondwana. Just to end my talk, I just wanted to introduce you to a wonderful site that we found. This paper is actually coming out quite soon. It's on Ouberg Karoo, and this is a new locality that we found. It is spectacular. It has the highest concentration of insects we've ever found in the country. and also diverse, and also, the preservation is absolutely stunning and amazing. So relative to Ouberg Pass, which is in this part of the escarpment, Main Karoo is on the southern part of, to just outside Sutherland. And this is the team we're excavating in that particular site. And obviously I was finding the best fossils over there. We have leaf whorls that we found, which will actually be good in the study that, with some of the research that I want to do in terms of comparisons. And we also have beautiful Glycopteris leaves. As you can see, there's wonderful insect damage on them. And this is the squamella. So if you look at the reconstruction that Miriam White has done, ours would be quite different from her, but not too extreme. But basically, we have cones of this plant, like cones of squamella, and they both most of them are fertile, and we have them in mats. So this is a huge slab in situ of squamella all over, and we have mats and layers of this fertile structure, which is actually a first ever. And this is a reconstruction of what Legitonia looks like. We have the very first cones of Legitonia that we have found in the world. So this paper is actually coming out as well. We're still trying to finalize a couple of things, and then we'll be submitting it. But this is a reconstruction that we did for one of our presentations, just to show you what it looks like. This is not the final thing, but this is just to show you what it looks like, briefly. And just to end, I'd like to thank our wonderful technical staff and volunteers, without whom this project would not be possible. And I'd also like to thank Fossil Friday Chats for inviting me to give my talk. You can get me on Twitter at Udamisha, and you can get me on Instagram as a paleobotany queen. And this is my email address, and this is some of my team members. So thank you very much, and I'm open for questions.
That was so cool.
Paleobotany queen indeed.
Every time we have like some cool like paleobotany thing, I just have like this cool thing where like I write down everything so that I can go read on it later, because I think we were talking about this earlier of how like we're so concentrated on vertebrate paleontology a lot in like the world of paleontology that sometimes we forget that everything is kind of connected, not to be too Disney, but like the animals and the plants are all connected, and that it's so important to really think about the plants and the environment and how that affected the animals and vice versa. And so it's just so fascinating to see research like yours. It's not something that I experienced very much in my career, unfortunately.
This talk was actually just, a tip of the iceberg of the things that I'm actually working on in terms of Glycopteris. But I just wanted to give you like a broad, insight to my world and to the things that I'm doing. So I just hope people are interested, and I'm open for questions if they are any.
I definitely am gonna be reading your paper for sure.
Yeah, exactly. Yeah, that's on my list. I'm gonna be very excited when that comes out. One thing I was thinking about, and you kind of said, in the beginning, like why study paleobotany? But I'm wondering what you think is the what when you do research into paleobotany, like what are you hoping to find? Like what kind of questions are you hoping to answer, like in terms of the environment or in terms of climate change? Like is there a specific like trying to find out more about this kind of part of the world through plants that you might not be able to find through animals?
Yeah. So I think it's different for different people because I mean my work is basically the building blocks is the foundation for everyone who's working on Glycopteris. So whether I say there are 5 species of Glycopteris or whether I say these leaves are all the same, it's basically the foundation for me, and then afterwards all the fancy and lovely work can be done. But for example, for me, I mean, in terms of working with in our time period, it's actually hard to do all these models of climate modeling and all the fancy things that, angiosperm people work with. I mean, I'm always, I always, whenever people ask me about, have you tried this? Have you tried this? I'm like, well, it's having analogs for the work, I mean, for the fossils that we work on. And I mean, angiosperm people can just walk outside and find, a living analog. I'm just being, but I mean, so we don't have any comparisons. I mean, these plants got extinct millions of years ago. I mean, the end of the Permian, which is the biggest extinction in the world. So it depends what your interests and your research interests are. So I mean, we're just doing, well, for example, now I'm doing the groundwork for basically hundreds of people to follow who will be doing work on Glossopteris and who do not want to work on the leaves, because no one has wants to work on the leaves. As you can see, it's a pain.
Somebody's got to do it though, and you're doing it well.
Someone's got to do it. Yeah.
I think that's an important point, is all of the, like, a lot of times when it comes to looking at paleontology from the outside, we see all the fancy studies and whatnot, and there's a lot of groundwork that goes into establishing that and to make that possible. And I think it's really important to do that kind of work, even if it's not, like the stuff that gets all splashed around.
Yeah, but I mean, like, for example, in South Africa, I mean, we're lagging compared to the other parts of the world. We don't have huge research teams and labs, where papers can just come out and people do certain things. We, like, as you saw, there are only 3 people who are paleobotanists, and you can't expect all 3 of us to look at the Permian, look through the entire time period in geological time period. So it actually assists, like, it helps when you have a lot of people in your team. But for example, in our team, we have people who are looking at various things. So we're basically trying to build this food web of of the Permian, of Gondwana. And this is a step forward. My research is just a small contribution to the bigger picture and the bigger goal of our team.
That's awesome.
That's amazing. I think what you're doing is such fantastic work, and I really hope people see it and get inspired to start working on those leaves and really bringing the amazing fossil collection, or the fossil record of South Africa, to the greater scientific community, because we can learn so much more about things that, like I said, I learned a ton today. It would be amazing.
Yeah, I mean, our research group is always looking for students, and I know my supervisor's out looking for a postdoc and PhD students who are, who would be interested in working on Permian floras. And so, if people are interested, just give us, a shout and say, hey, this is something that I'd be interested in actually working on and, contributing to the bigger goal and, the bigger records of South Africa.
Yeah. And with views like that you had, oh my gosh,
come on!
That beautiful.
It's
so
absolutely
gorgeous.
Stunning.
Yeah.
absolutely stunning. Yeah.
I'm definitely gonna set up a vacation for there real soon because I want to see that. That was just amazing.
Yeah.
Oh, definitely. I mean, like, we always welcome visiting researchers, people who are interested in knowing our work and, helping us, in the field to, get more material basically and learn as well. So,
All right.
give us a shout
We'll be
when
there.
you're in town.
Oh yeah, I'm definitely gonna give you a call.
Same. This is a good question from Dr. Alton Dooley. Considering all of the insects at the sites, do you often find Glossopteris with preserved insect feeding traces?
Oh, yes. I mean, I
Cool.
showed you a little glimpse, but I didn't show you everything. So I forgot to actually, show you the details from all the other leaves that I actually had seen, like I had showed. So there are people, like, for example, I do a lot of work on the taxonomy of the leaves and describing the leaves and the stats. And they also, in collaboration with my supervisor and one of the postdocs in our lab, we're actually looking at the insect damage. So there's lots and tons of insect damage, and they new insect damage types that we actually are describing. And this is also in the works. So one of the postdocs is actually spearheading, the research that we're doing in terms of insect damage, and they will be the one that will, they're working on the paper. So it's a team collaborative thing. So yeah, they're amazing. Yeah. That's
I love that. Not that only that you can find insects and plants, but you can also see the interaction in the fossil record between those two entities.
Definitely.
That's so cool.
Everything's connected.
Everything's
Yeah,
connected.
everything's connected.
I
mean, in some cases, you can even see where the insect laid its eggs. So like like the ovipositions where even failed ovipositions, like how like the pattern in which it was laying the eggs, and it's just amazing. And then you can also do a connection about which insects that you find in that site and which ones could have actually done the damage and which ones could have all these links that you can do. And yeah.
Oh, that's amazing. I'm getting kind of like, I'm just feeling so excited. That's so cool to be able to see something like that. I actually didn't realize you could see that kind of evidence in the fossil record.
Yeah, you can.
Oh, that is so cool.
That's cool. All right, to close out, since we're coming up on time here, I think this is a great question to end with. This is from Alexa, who is 9 years old. Hello, Alexa. Asking, what is your favorite fossil?
Glycopteris.
I like it.
Yes.
Just no hesitation. Like,
honestly,
the thing I've studied, that's it.
I think I love Glycopteris. I love everything about Glycopteris. And we're learning a whole lot of things about Glycopteris each and every single day we work on this, on this fossil. And Alexa, I do a lot of, you can ask your parents to actually, look on Fridays, Fossil Friday chats not chats, but like Fossil Friday, what's it, hashtag on Twitter.
The hashtag, yeah.
I do, yes, I do. There's a series I do with plant detective, fossil plant detective. And so if you can just follow those trends, and you will learn a lot about the different plant fossils that we find and the papers that we have in the lab, which is much more condensed and much more easier to understand for you.
Awesome.
That's awesome.
Britt, there was a last minute question from Dr. Paula Diaz.
Yeah, and we've got time for that. Are there insect coprolites in the leaves?
Not that I know of. So the thing is, there is so much. We have a lot of. We're still working through the material. I've photographed all the leaves in the material that we have, and because obviously there are people who are looking at the damage, and there's me who's doing the taxonomy and the measurements, and a whole lot of things. So in terms, we do have coprolites, like we do have identified coprolites, but I'm not sure of the specifics of the types of coprolites that we actually find there.
Awesome. Is that the last question, Brent?
That was it.
All right. So, tradition here on Fossil Friday Chats is we always ask one final question. What kind of advice would you give to a future paleobotanist on how to get into the field?
Just follow your dream and passion, and be the brightest star in the corner where you are.
Awesome. I love that. I actually want to put that on a t-shirt. That's so cool. Well, once again, thank you so much for joining us on the show. Thank you so much for sharing your love and passion and research. I know I have a brand new passion for Glossopteris, which I'm going to be reading about at lunch. And yeah, so thank you so much. Where's the best place that our audience can follow you, read more about your research?
You can follow me on Twitter. You can follow me on Instagram. So you can share my details in your YouTube thing at the end, and they were also in my presentation. So yeah.
Awesome. Well, thank you all so much. We'll be sure to put those links in the description below. As always, if you like this program and programs like it, want to support programs like it at the Albany Museum and Western Science Center, you can find links on how to do that below. And please be sure you like and subscribe to our channel for more stories from the world of paleontology. Thank you all so much, and we'll see you next week. Bye, everybody.